All right, good morning. So we're going to talk about the phospholipid bilayer a little bit, as well as moving in to talk about how the phospholipid bilayer is a piece of the cell membrane. So it's not just made out of the phospholipid bilayer, it's made out of actually quite a lot more than that. So we're going to talk about both of those. But in order to actually understand the cell membrane, we need to understand what its kind of foundational structure is, which is this, phospholipids. So phospholipids are kind of made out of two big parts. There's the head, which is hydrophilic, and there are two tails, which are hydrophobic. So the generalized phospholipid structure kind of looks like this. You have a phosphate group at the top, then you have a glycerol, and then you have two fatty acid tails. So the head of a phospholipid is composed of the phosphate and the glycerol. So this is an essential component because that's going to dictate how it is arranged. Because of the way that the phosphate works, it's going to be polar, right? And so that's going to change the way that the whole molecule functions, where the tails are going to be nonpolar. And so that's going to dictate how they function. Now here are the tails, so they are called fatty acids in general, and a fatty acid is just a chain of hydrocarbons, right? Hydrogens and carbons. So we call this a hydrocarbon chain. There's two of them, and this is a very simplified drawing of it. And interestingly enough, one of the tails is usually going to be saturated and the other is going to be unsaturated. Now, that's not true for all hydrocarbons, but in the case of the phospholipid, it is. So, saying that they can be saturated or unsaturated, you might need to do a little bit of review of macromolecules. Now, if you've never learned about macromolecules, you can kind of pay attention to the next couple of slides. So, saturated versus unsaturated. Saturated means that there are as many hydrogens as possible bonded to the carbons, right? So there's no double bonds. So you can see that this, the top line here, right, this top hydrocarbon chain is a saturated hydrocarbon chain. And what that does is it causes a straight chain, and that will decrease the fluidity of that fatty acid. And in this case, it'll decrease the fluidity of the membrane. Because when you have a straight chain like that, obviously you can pack them a little bit tighter together. Yeah, so imagine lining up a bunch of pencils that are straight, right? You have all these straight pencils, you can line them up pretty close together. But what would happen if you took a pencil and started to snap it, but don't snap it all the way, and it's kind of like bent, and you tried to line it up with all of those straight pencils? You obviously can't get all of them as close together. Okay, so that's kind of and not a great analogy, but an analogy for saturated versus unsaturated. So here's unsaturated. What this means is it just means that there's less hydrogen present than can possibly bond to the carbons. So that means that there has to be at least one double bond between the carbons. And what this does is it causes like a kink, like a bend in that fatty acid chain. And that can increase the fluidity because the kinks mean that you can't get them so close together. Okay, so here's kind of what it looks like. In the case of if you had two fatty acid tails, for example, you have the one on the left, that's your saturated, and they're nice and straight. With unsaturated, you could have both of them be unsaturated. You could have one be unsaturated and one be saturated, etc. So, as you can see in this picture, right, if you had lots and lots of kinks in your fatty acid tails, then it's going to be a pretty fluid membrane. If you have some kinks, but some completely saturated fatty acid tails, it's going to be less fluid. Okay? So, for phospholipids, usually we have one saturated and one unsaturated. But again, that is not as important as understanding why in a phospholipid you could have what the benefit is of having saturated or unsaturated fatty acid tails. Okay, so phospholipid bilayer. The outside of the cell, we know, is basically, so the exterior and interior of a cell are actually watery, right? So the outside of a cell, there's water, and on the inside of a cell, there's water. And so that's actually why the phospholipids organize themselves the way they do, because those hydrophilic heads will orient themselves towards the water, and the hydrophobic tails will try to avoid the water as much as possible. And what's amazing is that this emergent property arises where they will self-orient like this, right? When we talked about emergent properties, here's another great example of that emergent property, where if you dropped a bunch of phospholipids in a bucket of water, they would naturally arrange themselves this way. All right? Now, phospholipids are something called amphipathic. And what I want you to do is you can go into PowerPoint and you can click on this great definition of amphipathic. And I want you to think about why it is that phospholipids are amphipathic. Okay? All right. So here's another little kind of version of the cell membrane, specifically looking at the phospholipids. Okay? And remember that a phospholipid is just one of the many, many, many phospholipids in the cell membrane. Okay? And note that they are not made out of 3 tails, but only 2. Okay. And they are a bilayer, meaning there are 2 layers of them. All right, moving on, let's talk a little bit about the cell membrane structure itself. So I just really like this diagram. It has pretty much everything that you need to know about it, minus the fact that I don't see any cholesterols in it. Cholesterols that are in there. So we'll talk about those. All right. So what we're trying to figure out is, how is the structure of a membrane related to its function? Pretty much in biology, every structure is related to function, and so that should always be in your head. Not just to learn the structure, but learn what its function is. All right, so our big idea here kind of is that membranes are something called a fluid mosaic. Basically, it means that the proteins and other items can move within layers of the lipids. So we have the phospholipid bilayer, right? And that's the fluid part of the membrane. And then we have a mosaic of proteins that are embedded in that membrane. And the really cool thing about this new understanding of our membrane is that they're dynamic and ever-changing, very much like our understanding of, the cosmos. We used to think that it was perfect and unchanging. We used to think that about cell membranes as well, and now we know that's not true. So why do we call them fluid? So there's this really great video, you're welcome to click on it and watch it. It's just really kind of talks about the fluidity of the membrane and why it's called that. So phospholipid bilayers are flexible. They allow for cells to change shape, because that's kind of an essential component of being a cell, is that you have to adapt to the environment around you, right? Characteristics of life. The membrane lipids and some of the proteins can actually move laterally. Now, they don't move vertically, so they can't just pop out of the cell membrane. But they can move laterally within the membrane. And because the actual phospholipids are not bonded to one another, it means that they can kind of move and shift based on temperature and those kind of things. And the proteins actually will drift in those lipids. Now, they move much more slowly than the lipids, and there's some reasoning for that, and we'll get to that a little bit later. And then some of the membrane proteins are actually connected to the cytoskeleton within the cell, and so they can't actually move very far, but they can move a little bit. And so it looks like this. So you can see we have these blue guys are proteins. There's quite a few. Some of them have these carbohydrates sticking out on the end of them. We have our cytoskeleton down here, right? We have our phospholipids, and we have these little cholesterols embedded in them. So this is a cell membrane. So the proteins that make the mosaic of our fluid mosaic model, there's a couple different types. We have integral proteins, and we have peripheral proteins. Integral proteins will go through the whole membrane, okay? And they're permanently attached to the cell membrane. Peripheral proteins, on the other hand, are not embedded, meaning they're only attached to one surface. It can either be the exterior surface or the interior surface, but they're not embedded. They don't go all the way through. And so oftentimes they will attach only temporarily, and occasionally they'll attach to integral proteins. And we actually see that here. See this kind of pink one? So that's a peripheral protein attached to an integral protein. You can see integral. Can't really say anything about this one yet because we don't know. But if they go all the way through, they're integral. If they don't, they're peripheral. So here's another couple of examples. So here we have an integral membrane protein, right? It goes all the way through. And we have two peripheral membrane proteins attached to that integral protein. Now, the membrane-bound proteins, there's a couple different types. There's receptor proteins, enzyme proteins, adhesion proteins, recognition proteins, channel proteins, pump proteins, and they all do different jobs, okay? So receptor proteins will bind to specific molecules, yeah? And this is specifically to ones that are outside the cell, so like hormones. And what this will do is it will change something in the cell. So it will trigger something to occur within the cell. Now, these receptor proteins can be either integral or peripheral. So if I were you, while I was taking notes on this, what I would do is make like a little table of name of the protein, its job, and whether it's integral or peripheral. Okay? So it kind of looks like this, right? So we have this receptor protein, and there's some sort of thing like a hormone or other molecule that binds to it. And when it binds to that protein, it causes that protein to signal a chain of events to occur. Okay? So here's an example of one where our insulin receptor proteins. Insulin is the hormone that's released by our pancreas when our blood sugar levels are very high. And it helps absorb, it basically tells cells to absorb sugar to get it out of the bloodstream. And so insulin will bind to insulin receptor proteins, which then cause the cell to open and typically close open the already usually closed glucose transport proteins, because we don't want sugar just constantly coming into the cell, which we'll talk about when we do osmosis and diffusion. But sometimes we need that to occur. So this opening of the glucose protein will allow glucose to come into the cell. So here's what that looks like, right? We have glucose out here. We have insulin here. It binds to an insulin receptor protein, and that will tell the glucose transporter to open. Glucose will come in, yeah, and we'll break down that glucose as needed. Now, there's also enzyme proteins. So these will promote chemical reactions that will either build or take apart biological molecules, and these can be either integral or peripheral. So a great example of an enzyme protein is ATP synthase, which you've heard about in when we talked about mitochondria, that they create ATP, right? Well, this is actually how they do it. In their membrane, they have this ATP synthase, and what that does is it will synthesize ATP. Okay, so it builds ATP. What that does, the way that it does that is it takes a phosphate and it puts it onto an ADP, or an adenosine diphosphate. And we learned the word phosphorylate in the last class, right? That's what this is. So it's phosphorylating ADP. And again, it's located in the membranes of chloroplasts and mitochondria, because we need ATP for both photosynthesis and respiration. Okay, we'll do one more in this video and then cover the rest in the following video. So here's ATP synthase. We also have adhesion proteins. What these do is they anchor the cell to the inner cytoskeleton or proteins outside of the cell, as well as to other cells. So sometimes we need to anchor cells together, and that's what anchor proteins will do. These can be either integral or peripheral. An example of this is cadherins. It's a transmembrane protein, and what it does is when there's calcium, it will bind cells within tissues together to deliver that calcium. Okay. I think we can get one more in. Recognition proteins. So recognition proteins are basically tags. They're labels, name tags. They're often in the form of glycoproteins, which are just proteins with that sugar attached to the top. And these can be integral or peripheral. MHC proteins, or major histocompatibility complexes, they're proteins on the surface of our cells, and they belong to each individual. And so these MHC proteins will interact with our immune system to identify which cells are ours and which cells are not. This is what basically, when you get a fever or something like that, that's your body registering a foreign cell. And the way that it does that is with MHC proteins, okay? So we'll talk a little bit more about this. in class as well. Make sure you bring your questions to class, and I will see you